Chapter 3, Cells, the living units.
Okay, the topic of discussion is cell division, which the first part is interphase, which includes G1, S, and G2 phase. In the G1, it's mainly the growth. The cell is basically doing its normal thing by synthesizing proteins. If cells in this stage stop replicating, then they are said to be in the G0 phase. The following phase is S, meaning the growth and DNA synthesis, ensuring that the future cells will have identical copies of the DNA. Continuing on, the G2, which is the growth and final preparation for division, meaning that everything in there is proteins are being synthesized and moving to their correct location where they need to be, especially the DNA. In the G2 checkpoint, meaning that the cell is checking to make sure all the DNA is replicated so it can move on to the mitotic phase.
Okay, so this is the mitotic phase and the stages of mitosis. So starting out with our first one, we have prophase, which is where chromatin condenses into chromosomes, the nuclear envelope breaks down, and the mitotic spindle is forming, this being the mitotic spindle. Then moving on, we have metaphase, which is where there's a division of two nucleuses and the chromosomes become attached to the spindle fibers. And moving right along, we have anaphase, which is where chromosomes move away from one another to opposite sides of the spindles. So they're separating right there. And then down to telophase / cytokinesis. Telophase is like the last one of the mitotic phase. Cytokinesis is its own thing. So telophase is where chromosomes uncondense and go back to chromatin. The nuclear envelope reforms and the mitotic spindle breaks down. And then right here, this little cleavage furrow that's forming on both sides, the pinching, is where cytokinesis is happening. And cytokinesis is the formation of two cytoplasms splitting into two new cells.
So the topic is movement of materials across the plasma membrane. So here we have selective permeability of the membrane. So it allows some substances to pass while excluding others. So nutrients come in, which is a good thing. That's why we have the smiley face, because we want that. We keep the bad stuff, we keep it out, because we don't want that stuff going in. Osmosis, which is a passive transport that involves diffusion of a solvent, such as water, through a selective permeable membrane. Homogeneity refers to the ability of a solution to change the shape or tone of the cell by altering the cell's internal water volume. So you have isotonic solution, so the cell remains normal and water moves in and out. So you can see here in the diagram. Then you have hypertonic solution, which the cell loses water by osmosis and it shrinks. So you can see the cell here and then it's like shrivels up. And you have hypertonic solution, so the cell takes on water by osmosis and becoming bloated and bursts. So here you have the smaller cell, it gets bloated and then it bursts.
Okay, so here we have the cell. Here we have the cell wall and the cytoskeleton, which provides support and cell movement. Here we're going to be drawing the nucleus. So here we have our nucleus. The nucleus directs protein synthesis. Inside we're going to have the nucleolus. And these nucleolus, they produce the ribosomes. Okay, let me change my color really quickly. Okay, next we're going to be drawing the smooth endoplasmic reticulum. These are smooth because they don't have any ribosomes on them, and the smooth endoplasmic reticulum makes lipids, so it's like for lipid synthesis. And then we're going to draw this smooth rough ER. Okay, so here's the rough ER, and it's rough because it has the ribosomes on it. Then you also have 3 ribosomes around the cell. Obviously, I'm not the best drawer, as you can see through this. And the rough ER that makes the proteins. Okay, so next we're going to use the black marker, and these are going to be the peroxisomes. You can't see them, never mind. We're going back to the blue. Okay, so peroxisomes right here. And those metabolize the waste, so they're picking up the waste all around the cell. And then we have the lysosomes, even though they're in the same color, but I'm going to draw an X on them so you guys can tell them apart. So lysosomes, they digest the food. Okay, moving back to the green, we have the mitochondria. Okay, so here we have the mitochondria, and that produces ATP, which is energy for the cell. Go back to the blue. Okay. Right here we have the Golgi apparatus. So that one packages, transports, and modifies the cellular products. So think of it as kind of UPS for the cell. Then the last one, actually wait, no, that's it. That's it.
All right, that's the cell.
All right, so next we're going to be talking about membrane junctions. There's 3 of them that we're going to discuss. All right, gap junctions are the first one, and that is just communication between cells. Next, tight junctions, and these form waterproof barriers between cells. H2O barriers. And then next we have anchoring or osmosis, same thing. That these just create strong communications between cells. All right, so we're going to be talking about the fluid mosaic model next. The fluid mosaic model explains various observations regarding the structure of functional cell membranes. So right here, what I've already drawn out is the phospholipid bilayer, this being the entirety of the phospholipid bilayer. These are phospholipids, which are amphipathic. So right here we have water-loving part of our phospholipid and the water-hating. So hydrophilic, hydrophobic. And on either side of the phospholipid bilayer, we have cytoplasm and extracellular fluid, which is our watery environment. So we're going to draw some proteins. So as you can see, this protein here is stuck on the inside of, or embedded inside of the plasma membrane, and these are integral proteins. And then right here are peripheral proteins because they're stuck on the outside. And then right here is also a protein, but this one is called a glycoprotein. All right, and we're going to draw a little so the thing that I just drew right here is a glycolipid, and a glycolipid is anything with fat and carbohydrates. And then underneath that, and this little red thing right here, would be cholesterol, which stabilizes the phospholipid bilayer. Like I said, this right here, glycolipid. And one thing that I forgot to cover is that these proteins, they have some of their own functions, and that would be to transport material across the plasma membrane. They act as enzymes, act as receptors, cell-to-cell recognition, and intercellular joining.
Finally, we have our different types of RNA. First, mRNA, which carries genetic information copied from DNA in the form of a series of 3 base code words, each of which specifies a particular amino acid. Next, rRNA, responsible for reading the order of amino acids and linking amino acids together. Finally, tRNA, which reads the message of a nucleic acid or nucleotides and translates it into proteins or amino acids.